We are now reporting a new fluorescent protein CRISPR. So the protein can crystallize inside the live cell. So please look at this diamond, green diamond. So this is the crystal inside the live cell where many proteins are moving around so actively. So amazing. And then we discovered that the protein crystal was recognized as autophagic cargo and was engulfed into the lysosomes by selective autophagic process.
Crystals are highly ordered structures made up of individual units, such as atoms or molecules, arranged in a repeating lattice. Most crystals require specific conditions, such as a high concentration of molecules, to begin forming. Their growth is slow, taking place over days or even years, and requires a stable environment. The inside of a living cell is very active, with many kinds of proteins constantly moving around the cytoplasm. It is difficult to imagine that a crystal can form in such a setting. However, a research team from Riken Brain Science Institute engineered a fluorescent protein that rapidly self-assembles into large, well-formed crystals inside live cells.
I was so surprised when I first found the crystal within a living cell. The next surprise was that the crystal forms very rapidly.
Dr. Tsutsui isolated a fluorescent green protein from a coral that he collected on a diving trip to Okinawa in 2002. While engineering this protein to change its color from green to red when activated by light, he observed fluorescent crystals forming inside cells. The protein was named CRISPR, which is short for crystallizable and photoactivatable.
To a surprise, it's a very rapid process. So after the initiation of the crystal formation, so the growth is completed within a minute, so as far as we observed in our time-lapse imaging experiment. And then we are curious to know how the cell responded to the crystal.
The researchers used X-rays at the Spring 8 synchrotron facility in Hyogo Prefecture to determine the structure of the CRISPR proteins within the crystal. This analysis was done without isolating the crystals from the cells, the first time such a feat has been accomplished. A closer look at the cells containing crystals using an electron microscope revealed that the crystal was covered by a membrane, which turned out to be the lysosome, an organelle that engulfs and digests protein waste within cells. The crystal forms even when the lysosome engulfment is prevented by drugs, which means that the crystal formation is completed before it is enclosed in the lysosome, and not inside the lysosome. What triggers crystal formation is still a mystery, one that Dr. Miyawaki and his team are working on solving. They think that they will eventually be able to precisely control when and where the crystal forms. As the crystal forms, the CRISPR proteins that are initially uniformly distributed throughout the cell are rapidly incorporated into the crystal until no free proteins remain and the cytoplasm is clear. How the proteins are pulled together through the dense cytoplasm, which is filled with many proteins, organelles, and cytoskeletal elements, for now remains another mystery. This study brilliantly demonstrates that proteins can form crystals in living cells and also reveals that a cell can recognize big crystals to selectively remove them. How does a cell recognize crystals? Solving this mystery may shed light on certain diseases where abnormal proteins are not properly eliminated. It is thought that a deficit in abnormal protein removal from brain tissue causes them to aggregate and leads to brain tissue damage seen in some neurodegenerative diseases such as Alzheimer's disease and prion disease. Further understanding of CRISPR crystal formation and its removal from cells may shed light on the mechanisms underlying neurodegenerative diseases.